Okay, hey guys, welcome back to the channel. So in the previous lesson, we talked about DNA replication, which occurs in the interphase, and how it helps in mitosis. So now we talk about how DNA replication occurs, and we talk about protein synthesis, transcription, and translation, and what the structure of a DNA molecule is. So let's carry on with this lesson. This chapter is a bit more towards the memorization side, and it's a bit tough to understand in one go. So if you find it difficult to fully understand it, I recommend you to watch this video at least like twice. So first of all, you need to talk about the genetic code. And the genetic code is basically the sequence of bases which is present on the coding strand on the DNA molecule. And we use this acronym, which is please do not use. So I just came up with that. And you just take note of the first letters of everything. So P, D, N, and then Q, right? So punctuated, which means that the codon, which is a sequence of bases, it will always be starting with the start codon, which is AUG, and it will end off with a stop codon. And it's degenerative, which means that some amino acids, they can be coded for multiple with multiple different types of codes, and this results in a lot of codes being wasted because they're all coding for the same amino acid. It's advantageous to the genetic code because if there's a mutation, there's also many different combinations which will result in the same amino acid. So the protein formed in the end will still be about the same, and it's not overlapping, which means that the base, which is of coding for one amino acid, does not code for the amino acid which is, or does not take part in the codon which is ahead or behind of it. And it's universal, which means that in all living organisms, the sequence of bases which code for a single amino acid will always be the same. So now let's talk about the structure of the DNA molecule. So the most basic structure of DNA molecule is a nucleotide, and ATP is what we would call a phosphorylated nucleotide. So the nucleotide, it's made up of three different distinct features, which is a pentose sugar, which can be ribose or deoxyribose. And deoxyribose is used for DNA, and ribose sugar is used for RNA. And it can be made up of nitrogenous bases, so it can be adenine, guanine, cytosine, thymine, or uracil for RNA. And it has phosphate groups. And ATP, it's said to be a phosphorylated nucleotide because there can be varied number of phosphate groups. So AMP, which is adenosine monophosphate, it has one phosphate group, and ATP will have 3, and ADP will have 2. So it's a phosphorylated nucleotide because there's multiple phosphate groups. It's made of adenine as a sugar, and adenine and ribose sugar, and they act as energy source for energy requiring reactions like protein synthesis. So the structure of DNA molecule, it's, as we know, is made up of bases of different types of bases such as adenine, thymine, uracil, and these bases, which they can be classified into purine and pyrimidine bases. So purine bases are basically double ring structures. You don't need to know the structures, but you just need to know that purine is adenine, thymine, and uracil, ATU, and it has a double ring structure. And cytosine and guanine, they're single ring structures and pyrimidine bases. And during complementary base pairing, cytosine and guanine, they will form 3 hydrogen bonds with each other, and adenine and thymine, or adenine and uracil, will always form 2 hydrogen bonds with each other. So you can recognize which bases are present by the number of hydrogen bonds if they give you a diagram. So how does this entire DNA molecule form? So we know that in the 5th carbon of one sugar molecule, and then the 3rd carbon of another sugar molecule, they will form something called a phosphodiester bond. And this is strong covalent bonds with the phosphate group, which connects the nucleotides, and then this forms a sugar phosphate backbone. And then 2 polynucleotide strands, which are basically long strands of nucleotides connected to each other, they will lay side by side, and this must be done in an anti-parallel direction, meaning the one strand will run in a 3 to 5 direction, and then the other will run a 5 to 3 direction. And hydrogen bonds will form between complementary purine bases and complementary pyrimidine bases. And we also know about this concept of complementary base pairing, which means that adenine and thymine will go together and cytosine and guanine will go together. And for RNA, it will be adenine and uracil. And this polynucleotide strand, once the hydrogen bonds are formed, they're held together by hydrogen bonds, and then they will twist around each other to form the double helix structure. So over here, this image here, we know that one strand is going in the other direction and this other strand is going this way. And there'll be hydrogen bonds between the nitrogenous bases. And this is the sugar phosphate backbone with third carbon and fifth carbon. Okay, so we also need to talk about DNA replication. So we briefly talked about it in chapter 5 that DNA replication occurs during interphase, which doubles the amount of DNA molecules. So let's talk about how this DNA replication happen exactly. So this is DNA molecule over here. So the molecule will firstly unzip and unwind, and this is done by this enzyme called helicase. It'll break the hydrogen bonds between the nitrogenous bases, the complementary bases, and it will create something called a replication fork. And at the origin of replication, there's an enzyme called RNA polymerase, which will come and add RNA primers, this is done because you need to extend the chain by adding RNA nucleotides. So RNA primers are basically RNA nucleotides. This is because the enzyme DNA polymerase, which adds the DNA nucleotides, can only extend the nucleic acid but cannot start a chain by itself. So unzip, unwind creates a replication fork at site of origin, and then RNA primers are added by RNA polymerase. So let's carry on. So there's another enzyme called DNA polymerase. This will attach to the 3'N, and then it will synthesize a new DNA strand in the 5' to 3' direction. And DNA polymerase adds DNA nucleotides. And nucleotides are present in the cytoplasm, but the difference is that they are activated. That means they have additional phosphate groups. The reason why they have additional phosphate groups is so that once the bonds are broken, this produces energy, and then this energy is required because you need to form bonds during DNA replication, such as hydrogen and phosphodiester bonds. And then you also need to know that this happens. There's two strands. One is a leading strand, and the other one is a lagging strand. So the leading strand, it is synthesized continuously, while the lagging strand is synthesized discontinuously, and it will contain Okazaki fragments. These are DNA fragments, but the Okazaki fragments are separated by RNA primers. So there's an RNA primer here, and then the DNA polymerase will attach here, right? And then it will start synthesizing in the 5' to 3' direction. 5' to 3' direction. So start synthesizing a DNA strand this way. But then, for example, over here, as the replication fork will expand, so this will continue going. So this is synthesized continuously, but there'll be no enzyme running this direction. So another RNA polymerase will come here, attach the RNA primers, and then DNA polymerase will continue synthesizing this until it meets the next RNA primer, which blocks the further synthesis. So the Okazaki fragments are this over here and here, and then there's RNA primers which are in between it. Okay, so the RNA primers, they must be removed because they are of RNA nucleotides, correct? So they're removed by this enzyme called RNase H, and then they replace with DNA nucleotides, and this is by another DNA polymerase. So it's not the same. So you can just write it's another DNA polymerase, and then you need DNA ligase because these DNA fragments which are there, they're not held together. So they need to be attached together by DNA ligase. So DNA ligase will synthesize a single polynucleotide chain. Okay, I'll just give you 5 seconds to take in the information and then move to transcription. Okay, I hope you understood that. So if you didn't understand that, rewind a bit and watch it slowly and read the slides. And this topic is a bit tough, but it's not too difficult to understand once you piece everything together. So let's talk about transcription and translation. So these are important for protein synthesis. So what happens in protein synthesis is that RNA polymerase will attach to the 3' to 5' strand and at something called a promoter. So this is where transcription begins, and it starts to unwind the DNA strand. You also need to know what a sense strand and anti-sense strand is. So the sense strand is a coding strand and contains the genes. So mRNA must have the same sequence as the sense strand. So at the initiation site, the RNA polymerase will begin to synthesize RNA strand in the 5' to 3' direction, but you need to use the antisense strand as a template. So if it uses the antisense strand as a template, that means the RNA strand which is being synthesized is complementary to the antisense strand. So it will have the same sequence of bases as the sense strand, except the change in thymine for uracil. Okay, so let's carry on. So let's look at this image here. So this is the antisense strand, and this is RNA polymerase over here. So it will begin to synthesize it in this direction. So complementary base pairing hydrogen bonds are being formed. So over here, instead of thymine, take note that over here, uracil will be being added. So there is no thymine present in the mRNA strand. So the last part of transcription is that RNA polymerase will add the RNA nucleotides. They are present in the nucleoplasm as well. And the RNA nucleotides, again, they must be phosphorylated, or they have additional phosphate groups. So the phosphate groups, they're also broken to provide energy. And the breaking of these bonds do provide a lot of energy. And it's important because, again, we need to form hydrogen and phosphodiester bonds to form a single polynucleotide RNA strand. So eventually RNA polymerase will come to this site, which is called the termination site. And at the termination site, it will be exposed to a stop codon. And the stop codons on the DNA strand are ATT, ATC, and ACT. So once it is exposed, the RNA polymerase will leave the DNA strand. The mRNA formed will break off, and the DNA will zip back together. And now you have your original DNA molecule. You'll have an mRNA molecule as well. And obviously, the last triplet on the mRNA will also be coding for the stop codon. So the next stage of protein synthesis is translation, and this will happen in the cytoplasm. And as we know, and we have studied in our previous syllabus, and also in cell structure, that ribosomes are the site of protein synthesis. So now we'll talk about how exactly ribosomes catalyze peptide bonds and enable formation of polypeptides. So ribosomes, they are the site of translation. So the 5' end of the mRNA, they will come and attach to this small subunit of the ribosome. So the ribosome, which is made of rRNA, they have two subunits. One is the large subunit and the small subunit. And at a time, the mRNA will expose two codons into the larger subunit. So there's another molecule called the tRNA molecule, and this is also present in the cytoplasm. The tRNA molecule, it has special features, you would say. It has anticodon. So the anticodon are three bases which are complementary to the codon exposed by the mRNA in the small subunit. And it has an amino acid attachment site. So this tRNA molecule, each tRNA molecule will only go and bind to a specific amino acid. So the codon which is exposed is complementary to a specific anticodon. That means the mRNA will be calling for a specific tRNA molecule to come to the ribosome, and that tRNA molecule will bring a specific amino acid. So you could say that the codon brings a specific amino acid to the ribosome. Okay, so the tRNA molecules, they bring a specific amino acid to the ribosome, and the anticodon, and it can bind to the mRNA by forming hydrogen bonds, because the anticodon and codon, they have complementary bases present. Okay, and the tRNA molecule, it's basically an RNA strand, but you need to take note it is a single strand, but it can fold into 3D clover shape. And when this happens, there'll be hydrogen bonds, there'll be complementary base pairing. And the most important feature of them is that it has anticodon and an amino acid attachment site. And their function is to bring a specific amino acid to the ribosome. So let's look at this image. So this is the mRNA molecule over here. And at a time, it will expose three bases. And this is one codon. And this tRNA molecule, it will also have three bases. And this is the anticodon. So these bases over here, they are complementary to each other. So they can form hydrogen bonds holding the mRNA and tRNA molecule together. So this tRNA molecule, for example, it can bind to amino acid, and this amino acid is met. You don't need to know about the specific amino acids. So what does this mean? So example, this codon here, this is AUG, which means that AUG will code for this amino acid. AUG results in MET being called to the ribosome. So what happens is that there's this enzyme in the large subunit. So this is the large subunit, this is a small subunit. The large subunit, they have this enzyme called peptide transferase. This synthesizes the peptide bonds. So recall in biological molecules, the peptide bonds are synthesis of amide linkages. So this will synthesize the formation of peptide bonds. So, sorry, one second. So it synthesizes the peptide bonds between the amino acids which are attached to the tRNA and brought to the larger subunit. So the two tRNA molecules at a time can come to the larger subunit, and after the tRNA molecule after the amino acid will form a peptide bond, the tRNA molecule will release the amino acid and it will leave the ribosome. So the ribosome will move ahead to expose another codon. So another tRNA molecule will be called to the ribosome. So at a time, many ribosomes can bind to the mRNA molecule. So this is efficient because it forms something called polyribosomes, increases rate of protein synthesis. So multiple polypeptide strands are formed. And when the stop codon is shown on the mRNA, this is the termination site, and then the ribosome will leave the mRNA. So rRNA, as we have discussed, is what forms a ribosome, and ribosomes have large and small subunits. And mRNAs will bind to the small subunit, and the tRNA will bring the amino acids to the large subunit. And the large subunit will also contain this enzyme, peptide transferase, which is important in forming peptide bonds. This is spelled wrongly, so it's meant to be peptide bonds. So let's look at this image. So peptide bonds being formed here, tRNA will release its amino acid and leave. Ribosome will move ahead to expose another 3 bases. So another codon is exposed. So a tRNA molecule with a complementary anticodon will be called, and this will bring its own specific amino acid. So for example, this codon will code for this. So this peptide bond will be synthesized and a polypeptide will be formed. So why is DNA a stable structure but RNA is not? And this hydrogen bonding. So what is the significance of the hydrogen bonding in the DNA structure? So we need to know that there's a large number of hydrogen bonds which provide stability to the DNA molecule. This also enables the formation of the DNA double helix structure because the polynucleotide strands will be held together, then it will twist to form the DNA double helix. So this prevents action of enzymes and chemicals on the genome. And there's weak. Okay, so you could also say that the hydrogen bonds are weak, but it depends on context of what you're trying to say. So if you say that the hydrogen bonds are weak, you need to explain in what context. So this allows the bonds to be broken and DNA replication and mRNA formation to occur. So the large number of hydrogen bonds do provide stability. So this is in context to stability of the DNA structure, but in context of allowing DNA replication and mRNA formation to occur, you could say that the hydrogen bonds themselves are weak. So it also ensures that correct bases are added because complementary base pairing. And there's also strong phosphodiester bonds. And this contributes to the stability of the DNA structure. This is cut wrong as well. So stability of DNA structure. So let's talk about the differences in replication and transcription. So in replication, you're adding DNA nucleotides and DNA polymerase is used. Transcription uses RNA polymerase and RNA nucleotides. In replication, adenine pairs with thymine and mRNA is being formed in transcription, so adenine pairs with uracil. In replication, both strands will act as template strands, but in transcription, the antisense strand must only act as a template strand. This is because you want to achieve the mRNA code to have the same code as the sense strand, which contains the codes, right? The genetic code on it. At the end of replication, the double-stranded DNA molecule is produced again, but for transcription, a single-stranded mRNA molecule is produced. Replication helps in mitosis and meiosis during producing more DNA molecules. Transcription is protein synthesis, and replication requires an enzyme called DNA ligase, but in transcription that is not required. And try to recall what DNA ligase is used for. So DNA ligase is used to connect the DNA fragments, these Okazaki fragments, and the RNA primers which are replaced with DNA nucleotides, so they're not connected and they need to form the phosphodiester bond. So at the end of replication, a single polynucleotide strand is formed. And let's talk about the structure of DNA versus RNA. So DNA, we know that it is a double-stranded helical structure, but RNA is a single-stranded linear structure. DNA contains deoxyribose sugar. RNA contains ribose sugar in their nucleotides. Thymine base is present in DNA, but in RNA, uracil base. There's complementary base pairing in DNA, no complementary base pairing and no hydrogen bonds in RNA, but you need to take note that in tRNA, it is present. DNA is longer, but RNA could be shorter. And DNA, the polynucleotide strands run in both directions, 3 to 5 and 5 to 3, but RNA is only 5 to 3 direction. Because we also need to know that sense strand is also 5 to 3 direction, correct? Because sense strand and mRNA must be of the same form, because sense strand contains the genetic code as I've been repeating. So that'll be everything for this chapter. It is a bit heavy in terms of memorization and understanding the step-by-step mechanism of DNA replication and protein synthesis. So if it was a little fast, I would recommend you go ahead from the start and rewatch it and try to get a better understanding of this. Khan Academy does have a better video and more detailed drawing explanation. You could consider watching that as well, although some of that might not be exactly required for our syllabus. So let's just cover some question and answers really quickly. So this question says, which statements about tRNA are correct? Contains base pairing, contains hydrogen bonds, and is single-stranded. So I'll give you a second to think, and I'll explain this. Okay, let's think about what the tRNA structure is. Let's recall tRNA structure. So we know that tRNA is a 3D clover shape. It is a single-stranded RNA molecule, and since it's folded, it has complementary base pairing and does have hydrogen bonds because there's complementary base pairing. So in this question, option 1, 2, and 3, they're all correct. So the answer should be A. So let's talk about this question. You don't need to know about what these enzymes or proteins do because they're given the roles in the question itself. So let's see what the question says. Question says helicase, a protein, and its role, unwinding the parental DNA double helix. Topoisomerase breaks and rejoins the DNA strands. Single-stranded binding protein binds to separated DNA strands to stabilize them. And DNA polymerase, this will synthesize strands of DNA. Okay, so let's look at the options. So they're asking you to match the function with the protein. So you could pause here, read all the options, and I'll explain after that. Okay, so helicase unwinds the parental DNA. So if it unwinds the parental DNA, it's making the strands available as templates. Because if the strands are not unwinded or unwound, the bases are not exposed and it cannot be used as template strands. So this, okay, so let's talk about, we'll skip topoisomerase for now because this does not, you can't really exactly make a guess using the role. It's a bit difficult. So let's just talk about the others. So we know that DNA polymerase synthesizes strands of DNA. So we know that it adds DNA nucleotides. Okay, so it grows the polynucleotide strands or synthesizes the polynucleotide strand. And SSP or single-stranded protein, this binds to DNA strands to stabilize them. And if it binds to DNA strands, you need to understand that once the replication fork is created, the SSB protein will bind to both strands. And if it doesn't bind to them, the bases, they are complementary, so they'll form hydrogen bonds and it'll just wind back together and form the double helix. So it prevents the original strands from reforming and basically resulting back in the double helix structure. So I would say the answer would be D, because it's a bit difficult to guess what the topoisomerase role and function would be without prior knowledge. So I would recommend you just do guess and check. I mean, not exactly guess and check, but try to match the options and cancel out the options that don't make sense. So, for example, let's look at A. So adds DNA nucleotides to 3'N. So this is obviously function of DNA polymerase, right? So A is wrong. Even C, let's look at C. So adds DNA nucleotides to the 3'N. So this is for single strand binding protein. So this makes no sense. So A and C is out. So you could do that. So do the cancelling method for MCQ. It's very helpful. And let's look at this question. So which type of sugar and which type of bonds are found in DNA molecule? Type of sugar, deoxyribose and ribose sugar are both reducing sugars. And what types of bonds are present in the DNA molecule? There can be only two, phosphodiester or hydrogen bonding. Glycosidic is for sugar molecules and peptide is in protein synthesis. So A and C are definitely out, and B, type of sugar must be reducing sugar, so the answer is D. So let's continue with the next question. So which row is correct for adenine? Has a single ring structure, is a purine, joins to its complementary base with 3 hydrogen bonds. So this is what I was telling you about in the first slide, that you need to know about which bases are purine, which bases are pyrimidine, whether they are double ring or single ring, and how many bonds they form. So adenine, ATU is a purine, and purine have double rings, while C and G, they are pyrimidine, and they have single rings. ATU forms 2 bonds, and CG forms 3 bonds. So the answer should be C, because adenine, double ring, it's a purine, correct? And it doesn't form 3 bonds, it forms 2 bonds. So the answer is C. So what statements about nucleotides is correct? Adenine and guanine are pyrimids. ATP is a phosphorylated nucleotide. Purines have single ring structure. Uracil replaces cytosine in RNA. So let's look at these options. Adenine and guanine are pyrimidines. Absolutely not, because adenine is a purine and guanine is a pyrimidine, but adenine is a purine. So option A is wrong. ATP is a phosphorylated nucleotide. That is correct. But let's just look at C and D as well. Purines have single ring structure. No, they have double ring structures. And uracil replaces cytosine in RNA. No, it doesn't, because uracil will replace thymine in RNA, because uracil needs to bind to adenine. So this is the last question. I hope you found the question and answer section useful. Hope you understood the theory. If you have any questions, leave them in the comment section. If you have suggestions, do leave them in the comment section. Hope you find these videos helpful. Hope you understood everything in this. If you enjoyed the video, do consider liking, subscribing, and sharing them with your friends so more people can benefit. And as usual, I hope all of these videos have been quite helpful. And I'll see you in the next episode or the next class. And till then, best of luck studying, study hard, all the best.